Hello everyone. So in today's video, we are going to discuss this question which has appeared in 2012 Life Sciences GATE examination. This is question number 6 in the biochemistry section. So this question says that the pair of amino acid which does not undergo post-translational modification is. The four options are given: asparagine and histidine, tyrosine and serine, asparagine and serine, alanine and glycine. So let's see. First, let's talk about the post-translational modification. Okay, or we can also call them PTM. So what are these PTMs? So as the name suggests, post-translational modifications. So from the gene sequences, from the DNA sequence, via the transcription, we get our RNA. And via the translation, we get our protein. So after the protein is being synthesized by this machinery, further modification which takes place in this protein are called as post-translational modification. So these modification can change the protein structure. It can change the protein structure, its conformation, structure in the sense conformation, and if the conformation is changed, uh, along with the conformation, it can change the net charge, and due to the change in the charge, their interaction with other partner proteins can also be changed. Either it can be promoted or it can be inhibited. So interaction, okay, its protein's role in the signal transduction pathway, its localization, its activity. So all of these things depends on the post-translational modification which takes place in after the protein is being synthesized. Okay. So in this post-translational modifications, there are several types. Okay. So post-translational modification are important for gene regulation. As we know that apart from these points, we can also say that we have different organelles, different types of cells in our body, and all have same genome. All the cells are made of the same genome, but the function of each cell and organ differs from each other. So how the genome is being regulated? Suppose one protein which is required in the brain cell, and its function is mainly in the brain cell. So what if it starts expressing in the liver cell, in the skin cells, and in the muscle cells? Because in those cells, there that protein doesn't have any function. So that's why there is a need for gene regulation. So to regulate the gene expression and activity, these PTMs are important. So suppose one gene which is being functional in one of the organelle or in one of the organ, that gene can be suppressed, or that protein can be suppressed in another cell of our body via the post-translational modification. So that is why this post-translational modification plays very important role in gene regulation. Okay. So there are several types of post-translational modification, and the very common one is phosphorylation. Phosphorylation. So phosphorylation of any protein. So what it does, it changes the this post translation modification takes place in one particular amino acid residue in the protein. So phosphorylation takes place on any particular amino acid residue. So suppose one amino acid residue which is having no charge on it, and it undergoes the phosphorylation, then it will become negatively charged because of the negative charge in the phosphate group, and that charge now can alter the interactions, localization, its activity, and make it inactive. Anything can be done. So this phosphorylation, which is done by enzyme called kinase or phosphorylase, and in the reverse, another enzyme, that is phosphatase, that plays role in removal of the phosphate group. Okay. So it can be any ways. Either the phosphorylation form is active and non-phosphorylated form is inactive, or the non-phosphorylated form is active and the phosphorylated form is the inactive form. So that is the regulation on any different kinds of proteins. So phosphorylation takes place on the amino acid residue serine and threonine mainly, and tyrosine. Another modification is acetylation. So acetylation mainly takes place. So there are different types of protein that undergoes acetylation. For example, I would like to take here as an example of histone proteins, which are the positively charged proteins. So these histone proteins, which are rich in positively charged amino acid such as lysine and arginine, so the acetylation of histone takes place on these amino acid residues. So due to its positive charge, due to its positive charge, histone remain bound to the negatively charged DNA, and that's why it makes the structure more compact. That's why less transcription on those tightly bound or compact nucleosome structures. But due to the acetylation, the histone proteins from the positively charged, the positive charge being reduced due to the acetylation of the that brings the negative charge on the histone proteins, and thus the interaction of acetylated histone with the DNA molecules become reduced. That's why the nucleosome structure becomes less compact, and it becomes more loose, and that provides the active site for the transcription to takes place. So there are several sites of histone that gets modified. For example, H3K9 residue. As we know, our histone is made up of 4 subunits: H2A, H2B, H3 and H4. And H3, in H3 subunit, the uh, ninth residue, which is lysine, that gets acetylated, so that So in the acetylation, the ninth residue of H3 subunit of histone gets acetylated, and that makes that H3K9 is closer in the vicinity of the promoter, and that acetylation of histone makes it active for the gene transcription and further translation. So that those are the active mark of the histone, active transcription mark, gene transcription mark of the histone after acetylation. So there are several other residues as well. So H3K9 acetylation, also H3K4 acetylation. So these are the histone residues that gets acetylated. So other than that, acetylation, we have one more very common post-translation modification, that is methylation. So acetylation, the enzyme which carries the acetylation is the histone acetyl transferase, also called as HAT, and other one which is involved in deacetylation, that is histone deacetylase. Okay, so that is for the removal of acetylated residues. Methylation is carried by DNA, methylation is carried by methyl transferases. Methyl transferases. So methylation also, it can occur to this lysine and arginine residues. The positively charged residues can undergo acetylation and methylation. Okay, so as we saw that DNA acetylation was important for the activation, or it works as an active mark of transcription in the acetylation, while at the same position, if methylation takes place, it makes the gene, it makes the that locus inactive, or it is a repression mark. Okay, so H3K9 acetylation work as a uh, gene active gene mark, and H3K9 methylation worked as inactive gene mark. Okay, so that is how this PTMs regulate the gene transcription. Another very common one is hydroxylation. For example, proline. So we have hydroxyproline. Okay, so all these are different kinds of post-translation modification. Other than these common ones, we have several other kinds where addition of a saccharide residue, a saccharide to amino acid residue, that is glycosylation, addition of lipid and other complex moieties. So there are several different kinds of post-translation modification that can take place on the amino acid residues. So, so if we'll talk about these PTMs and where on which amino acid residues they take place, so the one which harbors OH in their side chain, which harbors NH ammonium group in their side chain, or amide group in their side chain. So if any amino acid side chain harbors these three, it can undergo the different post-translational modification. So for example, uh, for phosphorylation, we have serine. Okay, serine. So in serine, we have the side chain residue, this, and in threonine, we have the side chain residues. So here, and also other than that, we also have tyrosine, which also have hydroxyl or the OH group in its structure. So all these undergo post-translational modification, okay, of phosphorylation. So the OH group, these three harboring the OH group. So other we have, for example, we have aspargine. So its structure, where it has the side chain of CONH2. So this also can undergo post-translational modification. Other than that, other amino acids, for example, proline, which has NH2 in their side chain, which has this cyclic side chain where NH2 is accessible. So that can also undergo this post-translational modification. We have all these positively charged amino acid, for example, lysine. We have this side chain. We have the side chain where amino residue is present. Similarly, in the arginine and histidine structure as well, we have this side chain. So these can also undergo several acetylation modification in histidine structure as well. So there are also all these NH2 groups are present which can undergo the post-translational modification. So if any amino acid residue which is given to you which harbors these three in their side chain, so that can undergo the post-translational modification. So if we'll see our question, so the pair of amino acid which does not undergo post-translational modification is, so first one is the asparagine and histidine. So as we have already seen that asparagine harboring the CONH2, histidine harboring the NH2 group, that can undergo the post-translation modification. So this is also not true. Tyrosine and serine, which has the OH group in their side chain, this can also undergo the post-translational modification. Again, asparagine and threonine, which harbor CONH2 and OH group respectively, that also doesn't go any post-translation modification. Fourth option is the alanine and glycine. So if we'll see the side chain of these, so alanine has methyl group and glycine is the simplest amino acid. It has hydrogen as an R group. So these two cannot go post-translation modification. So the correct option would be this one, alanine and glycine pair, that cannot undergo post-translation modification. NPTEL reference. Thank you.